Hello everyone. I am Martin Blanchard. I'm a PhD student at the Hubert Curien laboratory. It's very fresh because I started my PhD on the 15th of November last year. So what I'm going to show you now is mainly the work I did during my master internship, because we will reuse it as a base for our future work during the PhD. So as it was said just before, the title of my PhD is this long sentence, but I will explain it a bit more right after. Before this, I want to stress that this is a collaboration between Hubert Curien Laboratory and Symbiose Laboratory, because it involves a lot of biology, which is a field I am completely new to, and so it is very interesting for me. And I hope that I will make it interesting to you too during this presentation. So let's start with a small example of what we tried to do during my master internship. We have two images. On the left here, on the right, sorry, we have an image of podocytes, which are human renal cells, and the right, those are also podocytes, but the difference is that whereas on the left they are normal, on the left we treated them with a drug that is called PAN, and which is known to be toxic for the cells. And just by looking at the images, we can see that the cells undergone a series of change, mainly on the actin filaments in red here. And the goal of the work was to provide a model that is able to identify the biomarkers, or to allow to identify the biomarkers of the toxicity and to quantify the toxicity of the drug. On a biological standpoint, this is what it is a representation of a podocyte. You can see that it has a very complicated structure, and this is what it actually looks like. And you see that there are those kind of tentacles, I could say, which are called pedicels, and by intertwining, they create filtration slits, and this is what provides renal filtration. And by looking at the shape of the cell, you can understand that if we modify the shape, if we modify the structure of the cell, the renal filtration, the renal function will be affected. So we went to study one of the components of the cell, the actin, mainly because it is situated everywhere in the cell, and this will help us on the image acquisition, sorry. And I'm kind of lost, but we focused on the actin on the following work. So this is for my previous work, but now that I am in PhD, we have different goals. First, if you remember the title of my PhD, and I'm sure you remember it because it is very short, there is some key points. First one is structural changes. We want to widen the scope of our study, for example, by studying more than just the actin and other cell components, and also to try to by trying to find relevant image descriptions of different cell components. Another keyword is diagnostic, because we want to go a bit further than just quantifying the toxicity of drug, and we also want to enable the diagnostic of pathologies based on objective criteria. And the last word is explainable, because deep learning models tend to be like black boxes, which we do not know the internal functioning. So we need to be able to interpret in terms of biological properties every our decision and analysis. So now that I have introduced my work a little bit, I will talk about the workflow of the processing we created during my internship. So first, if we want to analyze cell images, we have to acquire images of cells. So the way it is done is we cultivate two populations of cells, one that we leave alone, that is non-treated, and one that we treat with the drug we want to evaluate. In this case, it is PAN again. And what you see here on the right is a typical well from a culture plate. And there is a grid here that represents a zone of image acquisition, an area of image acquisition, and the resulting image on this zone is this one. So it's a very large image with a lot of cells, and you could think that it is very easy to obtain a big data set by in these conditions. But there are a few challenges that make it a little bit more complicated. First, with each culture, we try to recreate the same parameters, but we cannot ensure they are exactly the same. So comparing two different cultures is complicated and sometimes not very efficient or precise. So we have to study each data set separately. Second thing that we have to take into account is that cells in one population can react in kind of unintended way. For example, control cells that are non-treated can just naturally die. And so during our study, they can appear like they were deteriorated by a drug, although they're not. And also, it can be the other way around. A cell that was treated can completely resist it and look perfectly normal. So those two things were challenges we have to work with during the whole study. Now that we have our images, we can process them. So here is a little schematic view of the workflow. So with our input images, we perform segmentation to work on single cells, and we find a way to represent the cell structure in a form that allows us to perform interpretable analysis, and that allows us then also to quantify the deterioration of each cell in order to obtain a toxicity quantification or toxicity score for the drug. And also, this little dotted arrow here represents the fact that we want to go from the representation to the initial image, to always being able to come back to biological properties. So first, the segmentation. So why do we have to segment the images and work on individual cells? It is because of what I just said earlier. Cells all react a different way. So we are forced to perform a statistical study of the whole population of individual cells. So we went for the segmentation with a classical method, a marker-based watershed. And you can see the result here. There's the outline that is drawn. And you can see that it performs quite well, but it has some difficulties, especially here when the cells are next to each other, when the cells are connected. But globally, the result is satisfying. And also, we didn't have that much time to implement another method. So we went with this, which is satisfactory, to extract a cell representation, a useful cell representation. So we went with a bag of visual words approach. It is a traditional approach for texture characterization. And in our case, the texture, what it represents is a cell structure. So we extract the texture on small square patches like this on whole cells. And with all these little patches extracted on the whole cell, we create categories of structures that we call visual words. And so we can represent a cell by the sum of its visual words, by the histogram of its visual words. That's what's represented here. And the advantage of a bag of visual words representation is that it is very easy to interpret in the way that if, for example, our categories, our visual words we have only 3 of them, square, circle, or triangle. by just looking at the histogram, you can know that a cell is, for example, 50% circle, 20% triangle, and 30% square. So it's very easy to interpret. And also the fact that we extract those visual words on patches here on the image, we can go back from this representation to this one. Always, we want to stick to biological properties. With this representation of cells, we want to quantify the deterioration of it. And in order to do so, we perform a binary classification of the cells between two classes, one that is affected or deteriorated cells, and a class which is healthy cells. These classes have not to be confused with the cell populations because, once again, inside the population, there can be outliers. That's what's represented here. If we say that C1 is the class of affected cells and C2 the class of healthy cells, we know that some healthy cells can, some non-treated cells in red, can appear deteriorated naturally. And the other way around, this blue cell here represents a cell that was treated but did not react to the treatment. So in order to perform this classification, we used a SVM, linear SVM, and it tries to classify the populations by dividing the two of them with an hyperplane. So here it is a 2D representation, so we just have this line, and it tries to maximize the distance between the closest samples and the line. And in the end, the distance between those samples and the border, the dividing line, represents the deterioration of the cell. And the classifier translates it into a probability of belonging to one class or the other. So we can use this probability as an indicator of the deterioration. So that's it for the workflow of our model. And now I will show you a bit some of our results and analyze the data we have as the output of our model. First, we can perform an analysis on the visual words. And here what we represented is the relative difference between the mean histograms of the two classes. So for each class, for each cell, we have an histogram. And for each class, we computed the mean histogram to see which visual words are preponderant. And to compare the two classes, we computed the difference. And this is what is shown here. So if there is a positive value, it means that this visual words appears more in a cell that is in the cells that are considered healthy by the classifier. And inversely, a negative value means that this is a visual word that appears most of the time in an affected cell. So if we sort this out to see better which are the most important visual words, we can then isolate the two most significant visual words and go back on the patches on the cells to see which kind of structure these visual words represent. If we zoom in a bit on those images, these are mosaics of 100 patches that represents instances of the visual word. And here on the right, we have a meaningful visual word of a healthy cell. What we can see, is a striped texture, and this striped texture corresponds to actin filaments, to actin fibers, and which are totally normal in the cell that is healthy. Whereas on the left here, we have actin also, but it is not in the form of filaments. It was depolymerized by the pen, and as I told in the introduction, if the actin is modified, we probably have a modification of the structure of the cell, and thus we have a modification of the renal function. So by this visual word analysis, we can find biomarkers of the toxicity. So that's our goal number one that is fulfilled. But still, there is goal number two that is left, which is to quantify the toxicity of the drug. So to do this, we reuse our probability or deterioration indicator that is given by the classifier. And if we plot the distributions of those probabilities for the two populations, so here on the left, there's the distribution for the non-treated cells. So on the x-axis, we have the probability. Here on the left is 0% of being affected. And here, 100% chance of being affected. And on the y-axis, it's the part of the population that belongs to this probability range. So we can, well, I can because I can read the numbers, but I don't think you can here. But for the pan-treated cells, there are a bit more than 40% of the cells that the classifier considers to be at least 95% deteriorated or having 95% chance of being affected. And if we take the mean probability, it reflects the toxicity of the drug, the violence of the drug. For example, for PAN here, the median probability is 0.93. But if we take a drug that is less violent, such as heparin, for example, which is also known to be toxic, but less, we have different distributions where the median probability is lower and it's only 0.74. So this way, we can quantify the toxicity of a drug. We can give it a score, kind of, to reflect how violent it is for the cells. So to sum up a bit what we managed to do, we fulfilled our goals. We have interpretable descriptors of individual cells, which allow to identify biomarkers of the deterioration. And two, we are able to quantify the toxicity of the drug, and two, we are able to compare these toxicities between different drugs. But it is far from perfect. First limitation this model has is that it struggles with less violent drugs. If we go back to this heparin distribution, here we have the dose of heparin we used, which is very high in reality. And so if when we try to perform the same analysis with smaller doses, the model really struggles to reflect the toxicity of the drug. So that's the first limitation. And the second limitation that I already talked about a bit in the beginning is that our study focused only on the cytoskeleton, on actin filaments, and probably drugs also induce changes in other parts of the cell. So we probably have missed some changes that could have helped us. So for the future work during my PhD, we have plenty of ideas and ways to improve what we have already done. And the first thing we can try to improve is the segmentation. So if I showed you an example where our segmentation performed quite well in the beginning, here I chose one where it performs really bad. So here is our initial image, and on the right here is our method. And it really struggles in some cases like this when the interior of the cell is almost empty. Whereas a generalist algorithm for cellular segmentation called CellPose, based on UNets, performs really better. The force of CellPose is that it's generalist, so it can segment almost any kind of cell. So it could be useful for us if we want to study other kind of cells. And here is the result I show you is really how it performs without further training or without fine-tuning. We have also other ideas to work on to improve what we have. We could try to change our classifier and go with something more precise. We can also change the way of representing our cells and going for another approach than bag of visual words. Or we can also completely change our workflow and, for example, use deep learning to directly learn the representation space of our cells. And on a biology side, we also have ideas for the future. First, one of our objectives is to use a different cellular model. Right now. Before we were using podocytes, but now we want to study the blood-brain barrier, which is far more complex because it is not just one kind of cell. There are different cells that interact with each other, that are layered on top of each other. And so it will be, it is more difficult to cultivate, and it will be probably more difficult to analyze, but probably it will be also easier to see changes, because if one cell changes, it can it can propagate in cascade to one cell to each other. Another direction we can take is to try to quantify a functional perturbation induced not by a drug, but by a phenotypical change. And typically what we want to do in the end is to use Alzheimer patient serum and to introduce it in the environment of our cells. So instead of a drug, it's just a change in the environment, and it could, and the goal is to see if this change modulates the permeability of the blood-brain barrier, and it could serve to see if the Alzheimer patient serum modifies this permeability of the blood-brain barrier, and then deteriorates the brain, kind of. I'm no biologist, sorry, but the idea is this, kind of. And another direction that I did not write here is that we could also change the input images. We have to change our image acquisition workflow. We could, for example, use 3D images, because actin, one of the reasons we use actin is that it's present on the whole volume of the cell. But if we want to study some adhesion proteins, those are only present on the surface of the cell. So if we do not look at the cell at the right place, we can totally miss those proteins, and working in 3D could avoid this. We could also work with non-fluorescence image, like electronic microscopy, sorry, and stuff like this. Thank you for your attention.